Thank you. Can I have the master list back again? Number 5, pill to get rid of crud clogging the lysosomes. That sounds like Aubrey.
Unless John Schlondorn is in the room. Is he here?
Schlondorn?
Loser. Okay, right. So, as I spoke yesterday about, the general principle here is to identify new enzymatic approaches, enzymes in other organisms that are able to degrade things that our cells cannot degrade and that accumulate and eventually cause what I like to call late onset storage diseases, such as cardiovascular disease and macular degeneration. At the moment, we have actually ended up having that theme as the #1 project that my foundation is supporting, both in in-house research laboratory in Sunnyvale and also in a couple of labs in universities, one in Rice University and also at Arizona State. But the whole thing is going a good deal too slowly because there are lots of different potential target molecules to try to get rid of. For each of them, we have to go and look for a lot of different types of bacteria, for example, because each time you find one, you've then got to see whether you can find the enzymes that they're using. You've got to ensure that the enzymes are not toxic. You've got to make sure that they still work when you put them in a mammalian cell. And there's an attrition rate at this. So we need more high throughput identification of such strains and validation of their efficacy and safety, both in vitro and eventually in vivo. And it's certainly very straightforward to break that project down, as I summarized yesterday, into a number of stages, a number of milestones. And we're certainly looking at a situation now in which anywhere between $1 million and $20 million could be very easily spent in a very effective way over a period of 2 or 3 years to accelerate the process towards developing these extremely effective therapies that actually reverse the accumulation of these various types of problem and thereby actually cause diseases such as macular degeneration and atherosclerosis to regress rather than simply slow them down.
Excellent. Thank you. Now we have number 60, magic pill to fix the nuclear scaffold.
I guess that's me again. The nuclear scaffold, or the proteins supporting the nuclear envelope, have been implicated in a progeroid syndrome, HGPS, and also implicated in natural aging by the Mistelli lab at National Cancer Institute. Mistelli has an idea for a molecule that would patch the defective splicing site on the DNA. But I recently did a literature search, and I hesitate to say this publicly, but it looks like there's a fair amount of turnover of the nuclear envelope by the process of autophagy, which gets digested in the lysosomes. So it occurs to me, and this needs to be verified, that if we can fix the lysosomal crud problem that's preventing autophagy from doing turnover properly, we may automatically fix number 6, the nuclear scaffold.
Thank you for your concision. Bill Andrews on number 7.
Well, first, is Mike West here? Because I think
Mike
to do
day.
this subject justice, he should be here too, because we do have different opinions on these, and it's best to hear both. Why are you pointing this way? Okay, so I'll represent Mike too. Now, first I want to discuss the idea of is it worth making a pill and to turn on enhanced telomerase levels. And I've been struggling with this since I used to work at Geron Corporation 16 years ago. And there really isn't a good experiment that we can design to test whether or not telomerase inducer will work, except to make, except to make a pill, and then we can test it when we have it. All the kind of experiments that you can imagine using gene transfection experiments and everything are fraught with problems because just transfection alone can cause side effects, et cetera, and they're very hard to control. So in terms of the first part of this is the only way we're going to know if a telomerase inducer is really going to do anything useful is to actually come up with the pill. And that's essentially the major goal of our research, is just to be able to get the pill so we can answer the question. Another part of this is what are the side effects, especially the issue of cancer. This is where Mike and I are having some different opinions of this. Now, both of us, we've both read more than just the titles of papers. We've thoroughly investigated the papers. We've read more than just the press releases. There's a lot of chaos in the literature on telomerase, and there seems to be a big competition of everybody trying to publish on telomerase to get something said, and there's discrepancies even by the same authors. There's several papers that have come out. I think the last one actually was 2004, suggesting that when you put the telomerase gene into mouse embryonic stem cells and make a mouse out of that, it does increase the risk of cancer slightly. But those same authors have published since then saying the exact opposite. And there hasn't been much consistency in the levels there. But still, we don't know, and we're not going to know until we really come up with a pill. But my interpretation of all the really careful analysis of the literature is that I think that telomerase induction will actually decrease the risk of cancer, not increase the risk of cancer. And that's where Mike and I are disagreeing. I'm not going to argue against this. I just want to make certain his point of view is expressed here too.
I have a question here. What would be the fastest way to see if it will actually extend life? I mean, ignoring the cancer bit. I mean, in what model organism could one test that would give us some reasonable information? Because your presentation was about testing it in humans, and it seemed to me surely there has to be some shortcut that we could know sooner than that whether it will actually extend life, because I think there's a lot of doubt whether it's cause or effect.
I think the questions that both of you are raising can be seen as related to each other, and the need to have an aggregate functional trial. But
I'll just go quickly and say you don't necessarily always have to use human lifespan as your assay. If you give this pill to an old person and they start acting like a 20-year-old or even a 30 or 40-year-old, and they're starting out at 80, you don't really need to wait 20 years or 50 years to see how long they're going to live if it makes them healthier.
hey, wouldn't LSD make an 80-year-old act like they were 30 or 40?
What John just said, part of what I was going to do. Robert, you want to go next?
I have a brief comment on that. The two species that haven't been worked with extensively, although there are people working with them in the lab in Germany that was commented on at the AGE conference, they were working with Nothobrachius fusari, which was a 3-month lifespan fish. That's a vertebrate. All the systems that are in, vertebrates are essentially the same, with all difference to Michael working with Drosophila. But a lot of people say, well, so you lengthen the lifespan of the fly, so what? But if people would take you more serious, if you said we lengthen the lifespan of a vertebrate, and if you're talking about taking it from 3 months to 6 months, you have experimental results very fast. The only vertebrate that I know that has a shorter lifespan is a pygmy goby from salt water, and they're around Australia, and the problem is working with saltwater aquariums in a lab in the middle of Illinois or whatever is harder than working with the Nothobrachius fusari.
As always, we get a very creative idea from Robert Bradbury. Okay, unfortunately, we have to move on. The magic pill to delete the mutant mitochondria. Who wants that one?
I'll take it as brief.
John will take it.
First of all, mutant mitochondria are a problem in aging. They don't seem to be a problem in young people. And I strongly suspect that once again, if we get rid of the crud clogging the lysosomes, that autophagy will take care of most of the mutant mitochondrial problems. But in case it doesn't, it might be possible also to find some molecular tag that would make the mutant mitochondria more appetizing to the autophagy mechanism.
Yeah, I think there's pretty good evidence that actually the first part of John's suggestion probably doesn't work. I think that the fact that we see these clonal expansions, these preferential replications of mutant mitochondria at the expense of the non-mutant ones in cells is pretty good evidence that says that it's not an autophagy problem. However, the second part I think I absolutely agree with. There are various efforts that have been attempted to try to alter this selective balance and make the autophagy system preferentially get rid of the mitochondria that we'd like it to get rid of. And those attempts have rather fizzled out due to lack of interest and lack of funding. So those are certainly areas in which, albeit only initially in vitro on a time frame of a couple of years, but nevertheless, very specific milestones can be defined, and very promising bang for the buck can be developed. There's a group in Newcastle in the UK that have been working on using modified nucleic acids for this purpose. There are various other approaches that are of interest. So I think that's a very promising approach.
Thank you. I propose Steve Cole.